The FACS Desk's data management system interfaces directly with FACS flow cytometry instruments. It works rapidly and efficiently to organize, collect, store, and analyze data for FACS experiments. This introduction to FACS Desk was videotaped at the Becton Dickinson Immunocytometry Systems Laboratories in Mountain View, California. You are watching Keith Kelly, head cell sorter operator for BDIS. He is using FACS Desk to collect data for an experiment that was prepared by one of his colleagues. Keith looks pretty relaxed for someone running a 4-parameter FACS analysis. That's because FACS Desk is doing most of the work. Each time Keith delivers a sample to the FACS, he presses a key on the terminal so that FACS Desk will begin collecting data. You don't need to know anything about computers to run FACS Desk. Every function can be called up with simple 1 or 2 keystroke commands. The step-by-step tutorials in the second portion of this presentation will detail the use of each command and explain how to interpret the information shown on the terminal screen. During this portion of the presentation, however, we'll simply introduce you to what FACS Desk does and how it works. This way you will be familiar with each aspect of the FACS Desk system so that learning to operate it will be fairly easy. Keith is now delivering a sample to the FACS for analysis. If we could look inside the FACS optical bench, we would see individual cells flowing single file out of the FACS nozzle. As each cell passes into the laser beam, it scatters more or less of the laser light depending on its shape and size. In addition, if a cell has bound a fluorescent reagent or is intrinsically fluorescent, it emits fluorescent light in all directions. The amounts of scatter and fluorescence generated by each cell are measured by light sensors that surround the cell's stream. On the FACS 440 that Keith is using, there are 4 such detectors. 2 are currently adjusted to respond to red and green fluorescence. The other 2 are positioned to measure the amounts of forward scatter and large or obtuse angle scatter. Each time a cell passes into the laser beam, these sensors convert the amount of scattered or fluorescent light they detect into correspondingly large or small electrical signals. If Keith takes data from 20,000 cells in a sample, 20,000 sets of these electrical signals will travel along the wires connecting these sensors to the FACS console. The FACS console converts these analog signal levels into digital values. To collect 4-parameter data such as this, FACS Desk records a list of 4 digitized sensor readings for each cell analyzed in each sample. This permanent list mode method of data storage makes it possible to run multiple analyses on sample results obtained from a single data collection. While FACS Desk is collecting this permanent list of data points, Keith can watch a transient 2-parameter display of the data on the FACS console oscilloscope. Keith's oscilloscope is currently set to monitor the fluorescein and forward scatter channels. This means that every time the FACS console receives a set of signals for a cell, it plots a point on the oscilloscope reflecting the relative amounts of green fluorescence and forward scatter detected for that cell. Taken together, these dots create a pattern often called a dot plot. This plot shows that most of the cells in this sample scatter relatively little laser light. To obtain another view of the data, Keith switches the y-axis on the oscilloscope to monitor red fluorescence instead of forward scatter. The clusters of dots in this red versus green plot reveal at least 2 distinct cell subpopulations. One stains positively for both reagents, the other doesn't stain with either. These dot plots are useful for monitoring FACS working conditions and drawing broad conclusions about experimental results. However, the various FACS Desk analysis options offer much more versatile and accurate views of your data. For example, FACS Desk can later use the list mode data for this sample to recreate these dot plot patterns in the form of a printed contour map. It can also draw histograms to display single parameter data, or it can run integrations that compute the exact percentage of cells that fall into any subpopulation visible in a histogram or contour plot. Before we explore these analysis options, let's take a closer look at what Keith does to collect data for a sample. Right now, he's using the BD Rapid Sampler to deliver the contents of sample well F1 to the analytic stream. When the sample is flowing smoothly, he presses a key on the terminal to begin data collection. Earlier in the day, the investigator who designed this experiment entered a protocol into FACS Desk listing the cells and reagents in each of her samples. This flashing arrow points out that sample F1 contains human PBL stained with anti-Lu1 and anti-IgM. FACS Desk automatically uses this protocol information to label and catalog the data list for this sample. When the data collection is complete for sample F1, Keith moves the flashing rectangular cursor to indicate that sample F9 will be next. He then delivers the new sample to the FACS and repeats the collection process. While the oscilloscope monitors data from 2 of the 4 FACS sensors Keith is using, all 4 digitized sensor readings travel to FACS Desk through a special hardware interface that connects the FACS directly to a VAX computer. To understand how these 4-parameter cell data packages get labeled, stored, and later analyzed, imagine FACS Desk as a data processing microworld that you control from the computer terminals in your laboratory. In the FACS Desk microworld, there are facilities to handle every aspect of running an experiment. The facility which stores and labels the raw data as it arrives from the FACS can be thought of as a data warehouse. Inside the warehouse, each cell data package gets labeled with an experiment and sample number and receives a unique storage address so that it can be found for subsequent analysis. When Keith uses FACS Desk to analyze the results for this sample, the data package for each cell will be sent to the FACS Desk analysis machine. This microworld facility can analyze and display the list mode data in 3 basic formats. It can calculate the percentage of cells in a sample that belong to a specified subpopulation. It can produce histograms that graphically represent the distribution of data collected by any one FACS sensor, or it can produce contour maps that show the correlation of cell data collected from any 2 FACS sensors. When you operate FACS Desk, you don't have to worry about where the data is in the warehouse or understand how the analysis machine works. You won't see direct evidence of either of these facilities on your terminal screen. As far as you're concerned, they're in the sub-basement of the microworld. The only facility that you interact with directly is your FACS Desk itself. As a FACS Desk user, you'll receive a private desk with drawers that help you to run your experiments and keep track of your data. Each time you use your FACS Desk, the terminal screen presents you with a view of all the drawers that are stored at your desk. If you open a drawer, the screen shows you what's inside. There are 3 different kinds of drawers you can install onto your desk. Protocol collection drawers store the information that FACS Desk uses to label data as it enters the warehouse. For every new experiment that you run with FACS Desk, you'll need to install a new protocol drawer. In this drawer, you'll find a set of computer-style index cards labeled row A through Z. To prepare for data collection, you will fill in these cards by listing the parameters for which you will collect data and the names of the cells and reagents you will place in each sample. When you are ready to begin collecting data for an experiment, you switch your protocol drawer into collection mode. At this point, FACS Desk reformats the protocol information into individual sample labels that list the cells and stains you entered for each sample. When you collect data, you tell FACS Desk which sample you are collecting from. FACS Desk then associates the appropriate label with the data as it gets stored in the warehouse. Switching into collection mode will also cause an analysis drawer to appear on your desk. New analysis drawers are empty when they are installed onto your desk. Then, as you collect data for each sample, FACS Desk transfers the information from the sample label onto another type of card, which it places into the analysis drawer. These cards serve as a permanent record of the warehouse location of the data stored for each of your samples. When you analyze data, you use these cards to place your analysis requests. The third kind of FACS Desk drawer is called a book drawer. Book drawers are not used for running experiments. They simply store text information. Every desk comes equipped with a book drawer that stores an online owner's manual. There is one special drawer that can be found on every user's desk. It provides chronological, author, and title indices to experiments stored in a fourth element of the microworld, the Analysis Drawer Archive. Whenever you collect data from an experiment, you may make this data available to other FACS Desk users by placing a copy of the analysis drawer for your experiment into the archive. Conversely, you or any FACS Desk user can use the archive catalog to look up and retrieve primary data from any experiment that has ever been archived on your FACS Desk system. The archive is not used directly in running experiments, so for now we will move it off screen to make room for the final and most unique element of the FACS Desk microworld, the Magic Secretary. Every FACS Desk user is granted a part-time interest in this secretary. It serves as your hands, eyes, and voice inside the computer. It accepts the 1 and 2 keystroke commands you type on your keyboard and translates them into work orders for the microworld. It keeps the drawers organized on your desk, controls data collections, transfers jobs amongst the facilities, schedules printer and video output, and lets you know what's happening as your jobs progress. To run FACS Desk, you only need to learn a few commands for manipulating the drawers on your desk, and a few more that will allow you to communicate effectively with the Magic Secretary. Now that we've shown you a little bit about how FACS Desk is organized within the computer, let's return to the real world and watch the Magic Secretary's press-to-digital skills at work. Dr. Thomas Kipps from the Genetics Department of Stanford University has allowed us to document a multi-parameter FACS experiment that he is running at the BDIS laboratories. The experiment is part of a study examining the genetic mechanisms that control the proportions of lymphocyte subpopulations in human peripheral blood. In previous experiments, Tom analyzed PBL from 3 sets of identical twins. In each case, the staining patterns from one twin turned out to be barely distinguishable from the staining patterns of the other twin. In the study Tom is running today, he will analyze PBL samples taken from a set of identical triplets, Brooke, Christopher, and Byron Van Dyke. As Tom runs this experiment with FACS Desk, we will periodically return to our animated microworld to see what the Magic Secretary is up to.
So you see, it's very fortunate that you 3 could come down here today, because after having looked at a series of different twins and finding that among twins the patterns are very similar, it'd be very nice to find out how similar triplets are in these patterns. That would take us a long way in helping us to define these patterns as being under some form of genetic control. So actually we'd like to go out and get this experiment performed by, oh hi Ken, how you doing?
Good.
This is Dr. Ken Ault. He's our Associate Scientific Director at Becton Dickinson. And this is actually Byron, and this is Christopher, and this is Brooke.
Hi, nice to meet you.
And they've actually been very good to come down here today, and we're hoping to look at their B-cell subpopulations. It may be quite interesting to see what we can find.
Good.
So I'm going to have to go off into the lab and see if I can get things ready. I'd like to meet with you again after we've collected some of the data so we can go over some of the results. And
yeah, I like that.
That would be very interesting.
Ready to go down and donate some blood?
Sure.
Come on.
Okay.
As Ken took blood samples from the triplets, Tom entered a protocol for his triplet B-cell study into FACS desk. Tom will enter a description of each cell and reagent in his study, then he'll mark which samples will receive each cell and/or reagent. Later, FACS desk will use this protocol information to label and catalog the data it collects for his samples. Let's look inside the computer to see what the Magic Secretary does to help Tom prepare his protocol. To catch the secretary's attention, Tom presses a special key on his terminal that is labeled Insert. Insert is a generic command that is used whenever you want the secretary to insert a new drawer onto a desk or to insert new information into an existing drawer. If you give the insert command when all of the drawers on your desk are closed, the secretary will understand that you wish to add a new drawer onto your desk. To prepare a new drawer, the secretary essentially takes dictation at Tom's desk. It prompts him to indicate whether he needs a book drawer or a protocol drawer. The secretary then asks him for a title with which to label the new drawer. After Tom enters his title, Triplet B-Cell Study, the secretary presents him with a menu of FACS sensors. When Tom has selected the sensors he will use in this study, Texas Red, Fluorescein, Forward Scatter, and Obtuse Scatter, the secretary installs a new drawer on his desk labeled Triplet B-Cell Study. For ease of illustration, we have abbreviated Tom's experiment title to the word triplets. To begin entering his cell and reagent information, Tom opens the triplets drawer. Inside this drawer, the secretary has placed a set of protocol cards that can be used to define the contents of up to 312 samples, 26 rows of 12 samples each. Each row card is essentially a 12-column table with one column for each sample. The secretary has tailored these cards especially to Tom's experiment. The left-hand column of each table is divided into a series of subheadings, one for cells and one for each of the FACS sensors that Tom indicated he would use for this experiment. As Tom enters each of his cells and reagents, the secretary automatically lists them under the appropriate subheading. To enter a cell type or reagent, Tom once again uses the insert command. For each cell type Tom inserts, the secretary prompts him to enter information concerning the strain or patient name, tissue source of the cells, the treatment of the cells, and the number of cells he will place in each sample. The secretary will also give Tom a similar set of prompts for each of his staining reagents. As we join Tom out in the real world, he is responding to the secretary's prompts for his final staining reagent, Avidin conjugated Texas Red. Tom need only enter his description of each cell type and reagent once. The secretary takes care of duplicating the list onto each of his row cards. To complete his protocol, Tom indicates which reagents and cells he will pipette into each sample by placing marks in the appropriate sample columns on each row card. When he has marked each sample, he'll request a printout of his protocol which will guide him as he stains the samples. A printed FACS desk protocol provides a set of sample grids, one for each cell type and reagent in the experiment. The grids are marked to indicate the wells or tubes into which the items should be pipetted. When his protocol is complete, Tom picks it up from the printer and then goes into the laboratory to prepare his samples. He begins by pipetting the appropriate cells and reagents into each microtiter plate well. Tom has found that staining in microtiter plates rather than tubes is considerably more convenient and actually improves staining quality, particularly when handling large numbers of samples. After an appropriate incubation period, Tom spins down his samples. He then aspirates the supernatant, leaving a pellet of cells at the bottom of the well. Now he taps the plate to loosen the pellet and resuspends the cells in staining medium. Next, Tom will stain the cells with second step reagents and wash them again.
Hi, Tom. How's your timing?
Oh, pretty good, actually. I'm on the second to last wash. I'll be there in about 5 minutes.
All right. We're running the dye laser?
Yeah.
All right. I'll go get set up.
FACS Desk provides a FACS calibration routine which aids the operator in setting each FACS channel to a consistent standard. Proper use of this routine will assure that data collected from one session to the next can be accurately compared. To carry out this standardization, Keith is running a set of specially calibrated beads through the FACS. He is familiar with the dot patterns that the beads should generate, so he can bring the FACS into rough alignment by watching the oscilloscope screen. To fine-tune, he calls up the FACS calibration routine by typing the word calibrite on the terminal keyboard. The calibrite display reads out mean signal levels for single beads on each FACS channel. When the mean value generated by a channel is close enough to the target value for the beads Keith is using, the column of figures for that channel will appear doubly bright on the screen, thus indicating that the channel is adjusted to standard.
How are things coming here, Keith?
I think I just about have it aligned. I just need to focus a little bit on the fluorescence channel.
Yeah, I see you got it tuned up pretty well.
That's about it. Now we need to bring it up with the PMT voltage.
You want me to get it?
Please.
How's that?
Up just a little more. We're getting close. There, we've locked it in.
Looks pretty good?
Looks fine.
Let's get going.
Now that the FACS is adjusted to standard levels, Tom can bring his desk onto the screen by typing the word desk on the keyboard.
Where do you want to start?
Well, let's actually start at C1. That's where we have the unstained control.
All right.
To prepare his desk for data collection, Tom shifts his protocol drawer into collection mode and selects the appropriate label for his first sample. When FACS Desk goes into collection mode, the Magic Secretary really starts earning its title. For its first trick, the Secretary transforms the triplets protocol drawer into a collection drawer, while simultaneously installing a new analysis drawer onto Tom's desk. Both drawers will be labeled with the same title that Tom entered earlier for his protocol. To prepare for collection, Tom will open both drawers. Since the analysis drawer was just created, it is empty. The collection drawer, on the other hand, contains a rearranged version of the information Tom entered for the protocol. In switching a protocol drawer into collection mode, the Secretary compiles a separate label for each sample. These labels will be used to identify and catalog the data for each sample as it enters the warehouse. To indicate that he's ready to collect data for a sample, Tom uses a one-key command called execute. Like the insert command that Tom used earlier, execute is a multi-use command that the Secretary interprets somewhat differently depending on the kind of desk drawer being used. Since Tom is using his collection drawer, when he gives the command to execute C3, the Magic Secretary will show him the collection label for that sample. A collection label has three separate parts. On the far right side, the cell and sensor headings from the protocol are listed for reference purposes. This column cannot be edited and is the same for every label in this drawer. The center column lists the contents of the sample as described by Tom in the protocol. Note that each reagent is listed across from the fluorochrome with which it is associated. The leftmost column of each sample label is reserved for FACS users to tell the Secretary how many cell data points to collect per sample and how the amplifier gains are set for each FACS channel. In a moment, we will watch Tom instruct the Magic Secretary to collect data for 20,000 cells per sample. He'll then indicate that his FACS amplifiers are set to collect logarithmic data for both fluorescence channels and linear data for both scatter channels. Tom only has to adjust these figures for his first sample. The Magic Secretary will then use the same left-hand column for each successive label unless he indicates otherwise. To begin actual data collection, Tom will once again press execute. When the Magic Secretary receives the second execute command for a sample, it instantly copies the information from the collection label onto a card and places it in the analysis drawer. At the same time, the Secretary opens the FACS interface to start the data flow. While the data is being collected, the warehouse tells the Magic Secretary where the data for each cell is being stored. The Magic Secretary, in turn, keeps a record of these computer language addresses on the new analysis card it created for the sample. While all this is going on, the Secretary will also keep Tom's terminal posted as to how many data points have been collected and how fast the data is flowing. This helps Tom and Keith monitor the working conditions of the FACS. When the correct number of cell data points have been collected for a sample, the Secretary will close the FACS interface and await Tom's command to view the label for the next sample. This cycle will be repeated for each sample in Tom's experiment. Now let's watch the collection process from the outside.
Okay, now we're in the collection mode. Let's go down to C1 and begin.
First, Tom hits execute to view the label for C1.
Does our setup match?
Well, I have to check. What we'd like to do is to collect logarithmic data on fluorescein and Texas red. Do you have it set?
Yes, I do.
Okay. Looks pretty good, actually. Okay, I'm going to collect that.
To collect, Tom hits execute again. This time, the label information is transferred onto the top portion of a new analysis card, which appears as a gray line at the top of the screen. As you see, the gray line lists the cell type in the sample, the name of each FACS channel from which data is being collected, and the sample number. When the collection is complete, Tom moves the cursor to his next sample, C3, and again hits execute to view its label. Tom first checks the cell and stain labels for accuracy, then presses execute to start collection. Watch again as the sample label disappears and its information is transferred onto a new gray analysis line. Since the cells in this sample have been stained, the staining reagent is listed on the gray line in place of the fluorochrome with which it is associated.
Okay, we're finished collecting now. I wonder if we can move up to B3.
Okay.
That's an interesting pattern, actually.
While Tom and Keith continue the collection process, let's take a look at the options FACS offers for analyzing data. If Tom were to print out the data just collected for sample C3, it would appear as a mammoth table showing 4 sensor readings for each of the 30,000 cells analyzed. These readings are scaled from 0 to 250. As we mentioned earlier, FACS can run 3 basic kinds of analyses on this data. It can produce histograms that show the distribution of data from any one FACS sensor. It can produce contour maps which, like oscilloscope dot plots, simultaneously display 2-parameter data. Or it can run integrations to count the number of cells in a sample that have specified staining and scatter characteristics. Each type of analysis has its own purpose and value. Frequently, all 3 are used to provide different views of the data from a single sample. To create a fluorescein histogram, FACS counts and plots the number of readings in the fluorescein column that fall into each brightness range between 0 and 250. The resulting histogram shows the proportion of cells in the sample as a function of increasing green fluorescence. The bimodal distribution in this histogram reveals 2 cell subpopulations. FACS can run integrations to show us what percentage of cells fall under each of these peaks. We specify a range of values called a gate, or sometimes a window, and ask FACS to compute the percentage of cells within the gate. The result of this integration tells us that 47% of the cells in this sample are in the population that stained brightly for fluorescein. Although the peak representing the dull population is taller, it only contains 45% of the cells in the sample. To further characterize the cells in these populations, it would be helpful to see the relationship of this fluorescein data to the data collected for another parameter. For example, forward scatter. By plotting pairs of fluorescein and forward scatter measurements for each cell onto a grid, we can create a two-dimensional histogram in which populations of cells with similar measurements for one or both parameters appear as mountains in a landscape. The same populations that were indicated by peaks in the fluorescein histogram show up in this landscape as three-dimensional mountains. The position of these mountains on the grid suggests that the cell populations that they represent are made up exclusively of cells that scatter relatively little laser light. From this vantage point, however, we cannot tell whether the two large peaks in the foreground are masking data in the background. To find out, we could print or display views of this landscape from several perspectives. However, FACS avoids this cumbersome procedure by generating aerial view contour maps that show all features of a landscape from a single unobstructed vantage point. From this overhead view, we can see data that was hidden to us. The shape of the far side of these mountains show us that each population contains a significant number of cells with higher scatter readings. It is important to understand the relationship of a contour map to a data landscape. Imagine a set of parallel lines that circle the mountain range at specific intervals from the base up. Then imagine yourself flying directly over the mountaintops and looking down. As the contour lines project into a single plane, the resulting illustration is a two-dimensional contour map. FACS can draw 4 different kinds of contour maps from a single data landscape. Each map uses a different method to distribute contour lines. A density map is generated just like a geological survey map. The contours are placed onto the landscape in equal intervals from mountain base to topmost peak. Density maps sometimes give misleading views of flow cytometry data. A tall, sharp peak will always have more contours than a low, broad peak. Therefore, the number of contours in a region on a density plot will not necessarily reflect the number of cells in that region. For example, these contour lines, when viewed from above, give the impression that the tall peak on the left contains many more cells than the low peak on the right. Yet we know from our previous calculations that these two populations contain almost the same number of cells. FACS probability maps provide a more representative picture of the number of cells under each peak in a landscape. Probability contours are distributed so that an equal number of cell data points will fall between each pair of adjacent contours. In this example, the space between each pair of contour lines represents 10% of the cells in the sample. As you can see, the visual impact of the contours in a region of a probability map is fairly proportional to the number of cells in that region of the landscape. For this reason, Tom Kipps and his colleagues usually use this kind of map to display lymphocyte subpopulation data. To determine the exact percentage of cells in any region of a contour map, we can once again do some gating. If we define a gate for each dimension, FACS will count all the cells that fulfill these gating conditions. FACS can also produce logarithmic and area contour maps. The basis for generating these maps and the purposes they serve will be discussed in Part 2 of this presentation. At that time, we will review all of the FACS analysis options and offer some guidelines for selecting appropriate options for various kinds of research projects. Let's join Tom in the laboratory now as he picks up some scattered distributions from the printer and explains his analysis strategy to Ken Alt.
Actually, Keith queued these plots out of our scatter distributions, and they're quite nice. I had to leave the room during the data collection, so actually I kept the scatter gates fairly broad open.
Right.
And I think now we can do some nice gating on our cells. These large cells by obtuse scatter and forward scatter, they're our monocytes. I guess we can just gate those out.
Right.
The obtuse scatter does a fairly nice job of getting those delineating that population of cells. I think that represents about 10% of our cells, but I'd like to just home in on our lymphocytes here and see what these distributions are like. It should be pretty interesting.
Right. I think by gating on these and looking at IgM versus IgD, we should get some very interesting results. I'll be anxious to see what it looks like, Tom.
Okay.
See you later.
For each contour map, integration value, or histogram that Tom needs, he must signal the secretary to place a request onto the appropriate card in his triplets analysis drawer. An analysis card looks something like this on Tom's terminal screen. The gray section across the top describes the sample and the parameters that were measured. On the lower portion of each analysis card, the Magic Secretary keeps a permanent list of the analyses requested for that sample. The white line on this card indicates that Tom has so far requested one analysis on this sample. It is the contour map that we saw him use to set his gates. We can see that forward scatter was plotted on the y-axis, obtuse scatter on the x. Tom used wide open gates for both fluorescence channels so that the map would include data for all cells in the sample. Now Tom uses the insert command to add a new analysis request to this card. As we see, Tom has requested a probability contour map showing IGH-1A fluorescein data on the y-axis, IgM Texas red on the x. He has entered limiting gates for both scatter channels, so that this contour map will show data mainly for lymphocytes. Once Tom has placed requests for all of the analyses he needs, he closes his analysis drawer, exits the FACS desk system, and leaves the Magic Secretary to submit his jobs to the analysis machine. The Secretary then arranges for a copy of the data from each sample to be sent from the warehouse to the analysis machine. When the analysis machine has processed all the data for a sample, it sends the results to the Magic Secretary. The Secretary then routes the completed analysis to a printer or a video output system. Let's join Tom and Ken Alt one last time to see the results of the triplets experiment.
Well, hi, Ken. This is great. What we have is the B-cell distribution patterns for each of the three triplets. And we can see that here's one of them, and the next, and the next.
Very nice.
And we have actually virtually identical patterns for their B-cells. It's really quite remarkable, particularly when you consider how different every individual is from one another, as seen here.
That's very nice.
We're very fortunate to be able to have these sets of triplets and twins to compare.
It's good, very strongly suggested genetic control. I think that's very important. Congratulations.
Thanks.
When you know what FACS Desk can do, learning to operate it is straightforward. Most systems use the VT220 terminal shown here. The keyboard is grouped into three sections. Only two are needed to operate FACS the main keyboard on the left and the numeric keypad on the far right, referred to as the magic keypad. The main keyboard operates like a typewriter. It lets you respond to the secretary's prompts by entering text, such as experiment titles, reagent names, and so forth. A few keys also serve special functions. These are return, delete, line feed, and tab. The return key is generally used to signal that you have completed your response to one of the secretary's prompts and are ready to move on to the next one. The tab key is sometimes used in place of the return key. It calls up certain optional prompts. If you give FACS an incorrect command, pressing your line feed key will return your desk to the state it was in before the command was given. If you make a typing error when answering a prompt, the del key allows you to erase the mistake so that you can type in the correct entry. The magic keypad controls your private desk and allows you to give commands to the magic secretary. The symbols on the magic keypad labels will help you remember which functions are controlled by each key. To use FACS, you must first turn on the terminal and log on to the computer. Your system manager will assign you a password and show you the proper logon procedure. When you log on to your terminal, the cursor appears next to a prompt, such as the $ shown here. To enter FACS, type desk and press return. In a moment, your desk appears on the screen not as a three-dimensional desk, but rather as a list of the labels on the desk drawers. Each type of drawer can be distinguished by the format of its label. A protocol collection drawer label shows an experiment title and a tag indicating whether the drawer is in protocol or collection mode. Analysis drawers are labeled with the experiment title, the name of the sorter or analyzer from which the data was collected, a unique collection number, and the date on which the FACS analysis was run. Book drawers are simply labeled with a title. Let's take a look at the drawer labels on this desk. First on the list is an analysis drawer for an experiment entitled 3-color Li1, Li2, GK5.1. The flow cytometry instrument used for the experiment is known as flasher. The collection number for this drawer is 947, and the date of the collection was October 13, 1983. The next 4 drawers are also analysis drawers for experiments run on flasher. There are 2 drawers entitled Cultured PBL. The upper drawer is an analysis drawer. The lower drawer stores the protocol for this experiment. It is now in the collection mode. When all of the drawers on a desk are closed, the screen displays only the drawer labels and the cursor. You may think of the cursor as a pointer with which you focus the magic secretary's attention. There are 2 sets of commands that you must learn to operate FACS. The cursor commands let you position the cursor, thereby pointing to a particular drawer or card. The multi-level commands let you tell the secretary which procedures to perform in relation to a drawer or card. To understand the magic keypad commands, it's helpful to know a little about the structure of your desk. The drawers on each desk are actually a series of program modules that are grouped together as a logical tree. Each module, or drawer, is a separate branch of this tree. When you enter desk, the cursor is at the top level of the tree, and each branch appears on the screen as a label on the front of a closed drawer. Each branch or drawer has 3 levels to which you may move the cursor. The down-level command, labeled by the icon shown here, allows you to move the cursor from desktop level into any drawer and down to any level of the drawer. Sending the cursor down level into a drawer is equivalent to opening the drawer. The cards within are revealed on the screen. The levels within each drawer correspond to different sections of the cards described in Part 1 of this presentation. The up-level command moves the cursor 1 level at a time towards desktop level. Movement of the cursor within a single level of your desk is referred to as flat cursor movement. There are 8 flat commands. Each key is marked with an arrow to show the direction of cursor movement. The solid black arrows move the cursor 1 character or space at a time to the left or the right. The word left and word right arrows make the cursor jump one logical word to the left or the right. The small, solid black arrows located in the center of the keypad move the cursor up one line and down one line. The small arrows labeled 8 make the cursor jump up or down 8 lines at a time. Let's take another look at the top level of this desk. The terminal screen is a window that lets you view up to 24 lines of text. This desk is currently storing more drawers than can be displayed on the screen at one time. Using either of the downward flat commands, we can scroll the remaining drawer labels of this desk into the viewing window. Using the 8 flat commands in combination with the up and down level commands, you can move the cursor to locate and view the cards within any drawer on your desk. Notice that the up level and down level commands each share a key with one of the flat commands. In each case, pressing the key alone will give the flat command which is marked on the upper half of the label. To access the command shown on the lower half of a key, you use the Enter key. Enter serves as a function shift key. Pressing it alone does nothing, but pressing it before a multifunction key will alter the command given by that key. To give the up-level or down-level commands, first press Enter, then press the appropriate key. The remaining keys on the Magic Keypad are commands for the Magic Secretary. Most of these keys are multi-level commands. Their effect may vary slightly depending upon the position of the cursor when the command is given. The Insert command, shown in Part 1 of this presentation, is a multi-level command. When Tom gave the Insert command at desktop level, the Secretary responded by inserting a new drawer onto his desk. The second time Tom gave the Insert command, the cursor was at the lowest level of his protocol collection drawer. The Secretary then responded by prompting him to enter a cell or reagent description. Insert is therefore a generic command that allows you to insert new information into your desk. The multi-level Delete command allows you to mark unneeded cards or drawers for removal from your desk. The Delete command generally works as a toggle switch. Giving the command once will place a delete flag next to the unwanted drawer or card. Giving the command a second time will remove the flag. Flag drawers remain on your desk until you checkpoint. The checkpoint command is given by pressing Enter, followed by the letter C on the main keyboard. Like the up and down level commands, Insert and Delete also share keys with 2 of the flat commands. To give these commands, you press Enter before pressing the key. Execute and Select are also multi-level commands. In general, Execute instructs the Secretary to begin carrying out specified jobs in the microworld. The Select key lets you choose items from menus presented by the Magic Secretary. The remaining 2 keys on the Magic Keypad help increase the speed and efficiency of FACS Desk. The Repeat key re-enters the last command you gave. The Mark key allows you to put place marks in sections of your desk. In addition to these commands, there are several macro commands you must learn to run FACS Desk. A macro command is a chain of individual commands that are linked together and assigned to a key on the main keyboard. To call up a macro, you press Enter, followed by the key to which the macro has been assigned. There are 4 system macros. Enter C, described a moment ago, is the checkpoint macro. When you checkpoint, the Secretary places a copy of your desk into the long-term memory storage of the computer. Any work that is not checkpointed could be lost in the event of a power failure. Enter E for Exit, both checkpoints and exits you from desk. Enter H for Help brings the online manual to the screen. Enter T for Top sends the cursor from any point in your desk to desktop level. You may also define your own macros for frequently given sequences of commands. A user-defined macro is demonstrated in the analysis tutorial. To review, there are 16 Magic Keypad commands. The flat commands and the up and down level commands allow you to position the cursor. The 4 multi-level commands, Insert, Delete, Execute, and Select, tell the Magic Secretary what actions to perform in relation to the drawer or card next to which the cursor is flashing. Mark and repeat are convenience features to increase your efficiency in running FACSDesk. Lastly, the system macros allow you to conveniently call complex procedures such as checkpoint, help, and exit. It's not necessary to remember all of the keys and commands at once. It's more important to learn the command structure within each drawer. It would be a good idea to keep a magic keypad diagram handy while watching the rest of this video. In general, for every experiment that you run with FACSDesk, the first step is to create a new protocol. The information you enter for each sample in your protocol is used during the collection process to catalog data. The same information is also used to appropriately label each integration, histogram, and contour map that you request when analyzing your experimental results. To add or delete drawers from your desk, the cursor must be at desktop level. When you give the Insert command on the Magic Keypad, the Secretary prompts you to select the drawer type. To create a protocol drawer, hit Select when the cursor is flashing to the left of the word Protocol, then press Return. A new prompt appears asking you to enter a title of up to 40 characters length. For demonstration purposes, we'll name this experiment Sample Protocol. Before installing the drawer, the Secretary presents a menu of FACS sensors. To select an item from the menu, use the word right or character right arrow to position the cursor next to the desired sensor. Then press Select. An asterisk appears next to each selected sensor. Select is a toggle key. If you choose a wrong sensor by accident, you can unselect it by pressing Select a second time. You must select every sensor that you think you may need for an experiment at this time. It is not possible to add additional sensors after the Secretary installs the new drawer. Watch again as we choose the sensors for this sample protocol. Using the Select key, we choose Fluorescein and Texas Red. Then, with the word Right key, we scroll past the next 3 sensors in order to select Forward Scatter and Obtuse Scatter. Now that we've chosen our sensors, we press Return, and the Magic Secretary instantly installs the new drawer. It is in protocol mode. To open the drawer, we use the command Down Level. This moves the cursor to the first or row level of the drawer, where you can see the top section of each row card. First, you'll see the card for row A. To view more of the cards, move the cursor lower on the screen with a downward flat command. Giving the down level command once again moves the cursor to the cell and sensor level of the protocol drawer. At the second level, the subheadings for entering cells and reagents appear on each card in black writing on a gray background. Again, you can move the cursor with the flat commands to reveal the cell and sensor subheadings on each successive row card. Sending the cursor to the lowest level of the drawer reveals a white line below each cell and fluorochrome subheading. This is the pipetting line level of the drawer. Each cell and reagent that you insert will receive a separate pipetting line on each card. Since this protocol has not yet been filled in, the pipetting lines are blank. The vertical flat commands move the cursor from card to card. To bring the cursor back to desktop level, you can give the up level command 3 times, or to save time, you can use the Enter T macro, which brings the cursor to desktop level in one jump. Cell and reagent information may only be inserted at the pipetting line level of the drawer. With the cursor flashing next to your protocol drawer, go down level 3 times. The cursor lands on the white pipetting line below the subheading for cells on the card for row A. To enter information on a cell, press Insert. The secretary will then prompt you to enter a cell strain or patient name, a tissue, organ, or culture source for the cells, a description of any special treatment or condition such as trypsinization or age, the number of cells in the sample, and the dilution volume. When you press return after answering the final prompt, a listing of the cell you described will appear on a pipetting line. To add another cell type to the list, press insert again, and the prompt cycle will repeat. In this example, we'll answer the prompts for mouse spleen cells. We type in the strain, BALB CN, then press return. The tissue, spleen. For treatment, the age of the mice, we select 1 million cells per sample in a 50 microliter dilution. When we press return after the final prompt, the secretary adds the new line below the first pipetting line we inserted. Fluorochrome reagent information is entered in the same manner. First, use the flat commands to position the cursor on a white line below the appropriate fluorochrome subheading. Then press insert and answer the secretary's prompts. The secretary will ask for a reagent name, the amount of protein in micrograms, and the dilution volume in microliters. The secretary recognizes the @ character as an abbreviation for anti. When you press return after answering the third prompt, the staining reagent appears on a pipetting line below the sensor heading. Now we'll add a few more reagents to the fluorescein list. Obviously, this sample protocol couldn't be used for a real experiment. The cells and reagents have been chosen to show examples of different types of answers to the prompts. To enter the Texas Red reagents, we position the cursor on a white line below the gray subheading for Texas Red, press Insert, and begin the process again. If you are collecting Texas Red data, it is common to run a two-step staining process using anti-IGH conjugated biotin and avidin conjugated Texas Red. The secretary recognizes two-step stains and will later on use the appropriate reagent name to label your analysis outputs. In this case, the secretary will label any printouts with IGH6A. When you insert a pipetting line onto a card, the secretary simultaneously inserts the same line onto every card in the drawer. Using the flat commands, you can move the cursor to reveal pipetting lines on any card. Here, by moving the cursor down one line at a time, we scroll through row B and now C. As the final step to filling in a protocol, you mark which samples in each row will receive each cell type and/or reagent. To mark a well, you can use the Select key, or you can press any letter on the main keyboard. Using the main keyboard not only marks the well, but also moves the cursor forward one space. To select samples in row A to receive CBA thymus cells, we position the cursor on the row A pipetting line labeled CBA thymus. We then use the character right key to position the cursor and press a key on the main keyboard to mark the wells. We have now marked all odd-numbered wells in row A to receive CBA thymus cells. To mark these cells for staining with anti-Li1, we press return or line down until the cursor is on the white line for anti-Li1 and mark the wells for this reagent. For example purposes, we'll complete the pipetting map for row A by marking wells A1, A3, A9, and A11 to receive the avidin conjugated Texas Red reagent. When we finish marking wells for row A, we use the flat commands to slide the cursor down to row B so that we can begin marking wells for that row card. If you place an asterisk under the wrong well, you can remove it by using the Del key on the main keyboard. If you wish to erase all of the marks on a pipetting line, use the magic keypad delete command. When you have finished marking wells, FACSDesk will print out your protocol as a pipetting map to aid you with your laboratory benchwork. To request a protocol printout, place the cursor next to your protocol drawer and press Execute. The word Working will flash, indicating that the secretary is putting your request on the printer queue. On the protocol printout, the name of each cell and reagent appears above a sample grid. Marks on the grid indicate which sample wells have been selected to receive the cell or reagent. When you collect data with FACSDesk, you switch the protocol drawer for your experiment into collection mode. This reformats the protocol information into a set of individual sample labels while simultaneously installing a new analysis drawer onto your desk. As FACSDesk collects data for a sample, the secretary transfers the information from the collection label onto an analysis card which it places into the new analysis drawer.
Okay, take two.
To collect data, you must first log on to the terminal that is assigned to your flow cytometry analyzer and call up the FACS calibration routine by typing the word calibrite. Using calibrite to standardize each FACS channel assures that data collected from one session to the next can be usefully compared. Instructions for calibrating the FACS can be found in the calibrite manual. It's not required that you complete the calibration routine, but you must at least call for calibrite before the secretary can accept the command to enter collection mode. To exit calibrite, hold the control button down and press the letter C on the main keyboard. Then type desk to bring your desk back on screen. Enter execute as the command for switching a protocol drawer in or out of collection mode. We'll now toggle our sample protocol into collection mode. We position the cursor next to the drawer and press enter execute. As the drawer switches modes, a new analysis drawer appears bearing the same title as the protocol collection drawer. The analysis drawer is easily distinguished from the collection drawer. Its label shows a facility name, an identification number, and today's date. After switching into collection mode, the next step is to select and view the collection label for your first sample. With the cursor next to the collection drawer, give the down level command. This sends the cursor to row level. The three levels of a protocol collection drawer are the same in both modes. However, the command structure is different. In the protocol mode, the secretary only accepts commands given at the lowest or pipetting line level of the drawer. In collection mode, on the other hand, the secretary only accepts commands given at the highest or row level of the drawer. Only in collection mode may the flat commands be used to move the cursor around the sample matrix. As the first step to collecting data for a sample, you position the cursor at the appropriate sample number and press Execute to bring the collection label on screen. Watch again as we press Enter Execute to toggle collection mode, then send the cursor down level into the collection drawer. You may collect sample data in any order. We will start with B3. Pressing Execute reveals the label. As you may recall, each collection label is divided into three columns. The leftmost column indicates how many cell data points will be collected and to which level each FACS channel is adjusted. The center column lists the cells and reagents that were marked for pipetting into the sample. The right-hand column lists the cell and sensor subheadings of the protocol. This column cannot be edited. If a sample label is inaccurate, it should be corrected before you collect data. To change any item in the left column, position the cursor, then use the character left and character right keys to view the available options. Watch as we scroll through the options on the cell line. The character left key decreases the number of cells to a minimum of 1,000. The character right key increases the number to a maximum of 100,000. The number of cell data points that you choose to collect depends on what type of analysis you're running. If you're running a 4-parameter analysis, 50 to 100,000 cells should yield enough data to reveal 2 to 3% subpopulations. Experiments which analyze fewer parameters require fewer cells. For controls or titrations, between 2 and 5,000 cells should yield ample data. A copy of this chart can be found in your owner's manual. We'll select 20,000 cells. Pressing return 3 times brings the cursor to the amplifier setting for the forward scatter channel. Again, we scroll through the options using the character left and character right keys. Selecting the appropriate setting for each FACS channel depends on the type of data it is measuring. The logarithmic setting is usually used for fluorescence channels because fluorescence data tends to vary widely from cell to cell. Other types of analyses, such as scatter or DNA staining, generate sensor readings within a much less dynamic range. Hence, it's more effective to set these channels to collect linear data. If you set a channel to the off position, the secretary will not collect any data from that channel. When setting the left column of a label, you must either adjust the actual FACS output levels to match your label, or else adjust the label to match the FACS. If they do not match, the scales on the analysis outputs for your samples will be incorrect. To save you time, corrections entered in the left column of one sample label are simultaneously entered onto every other label in the drawer. When the values in the left column of the label are correct, check the center column for accuracy. If there are any errors in the center column of a label, move the cursor by using the word right key. Then, with the arrows flashing next to the incorrect cell or reagent name, simply type in your correction and press return. When the sample label is accurate, you can deliver the appropriate sample to the FACS and press Execute to begin data collection. Let's review what happens when the secretary receives the second Execute command for a sample. First, it transfers the label information onto an analysis card and places the card into the new analysis drawer. Then the secretary opens the FACS interface so that data will begin flowing. While the data is being collected, the warehouse informs the secretary of the warehouse address for each data point. The secretary, in turn, keeps a record of these computer memory addresses on the new analysis card. When the correct number of data points have been stored for a sample, the secretary closes the FACS interface. Watch the screen now as we press Execute for sample B3. As soon as the white label box disappears, you know the magic secretary has started collecting and cataloging data. The new analysis card appears as a gray sample line below the analysis drawer label. The sample number can be found on the right edge of each sample line. When the data collection is complete for a sample, the sample number glows brighter on the matrix. Now we press Execute to view the label for sample B1. The machine conditions are already set for this label, so we simply press Execute again to begin collection. During data collection, the screen is very active. Arrows flash by the gray line for the sample being collected, the cursor flashes at the sample position on the matrix, and at mid-screen the magic secretary flashes a running total of how many cell data points have been collected and informs us of the rate of data collection. Watch the right side of the screen now as we collect data for sample B5. When the correct number of data points have been stored in the warehouse, the magic secretary displays the total, then goes back to the collection ready mode. We have now completed the collection process for 3 samples. We see only the gray top portions of the 3 analysis cards because the analysis drawer is only open to its first level. Now we'll go to row H, sample 1. If you execute a well to view its label, then choose not to collect data, you can press line feed to cancel your first execute command, thereby making the label disappear. If you have a technical problem while collecting data for a sample, you can abort the collection by pressing Enter Execute. There is no need to worry about collection interruptions. You may collect up to 7 times using the same sample label. Each successive analysis card will receive a new letter, for example, H3A, H3B, etc. If you need to check the FACS alignment during a collection session, give the command Enter Execute to bring the Calibrite display on screen. Control-C will return the cursor to its position before Calibrite was called. To terminate a collection session, move the cursor to desktop level and press Enter Execute. This will toggle the drawer back to protocol mode. Watch the collection process one last time. Here we'll collect data using a label that has been used once already, H3. We press Execute to view the label and press Execute again to begin collection. Notice the lowercase letter A, which distinguishes this data set from the previous data set that was collected using the H3 label. To terminate this session, we send the cursor to desktop level and press Enter Execute. This toggles the collection drawer to protocol mode and permanently seals analysis drawer number 4144 against receiving new cards. Toggling the sample protocol drawer into collection mode again creates analysis drawer number 4145. As we execute to collect data, the new card gets stored in the new drawer. When you are collecting data, it's a good idea to checkpoint every once in a while by pressing Enter C. Until you checkpoint, your data is not permanently recorded. Hence, if there is a power outage or some other problem with the system, you could lose valuable data. Unless you have some reason to keep your data private, it's a good idea to archive the analysis drawer for each experiment as soon as you have completed your data collection. That way, in case you accidentally delete an analysis drawer from your desk, you can always ask the magic secretary to retrieve a copy of it from the archive. To archive an analysis drawer, place the cursor next to the appropriate drawer label at desktop level and give the command Enter Execute. When drawers are placed in the archive, they are automatically cataloged and cross-referenced according to author, title, and date. The indices in the archive drawer are organized like a card catalog in a library. We will demonstrate the use of the author index to find and retrieve an experiment that was archived by Dr. Thomas Kipps. First, we must flag the drawer for retrieval. To open the archive catalog, we place the cursor next to the word archive at desktop level, then give the down level command. This reveals the index level. Going down level into the author index reveals an alphabetical list of investigators. Using the flat commands, we position the cursor next to Kipps. Going down level to the lowest or card level reveals a separate white line for every analysis drawer that Tom Kipps has archived. We place the cursor on the line for the experiment we were looking for, anti-human Ig hybridoma staining, and press Select. This marks the drawer with a fetch flag. To retrieve the drawer, we send the cursor to desktop level and press Execute. The magic secretary then copies the flag drawer and installs the copy on the desk. This drawer may now be used in the same manner as any other analysis drawer on the desk. For each histogram, contour map, or integration you wish to request from FACSDesk, you must insert an analysis line onto the appropriate sample card in the analysis drawer for your experiment. There is no limit to the number of analyses you may request for a single sample. To illustrate this section, we will use the analysis drawer from an experiment on mouse spleen cells performed by Dr. Paul Layler of Stanford University. Giving the down level command opens the drawer, revealing a set of gray sample lines, one for each sample in the experiment. Going down level a second time reveals a blank white analysis line on the card for sample A1. To request any type of analysis, press Insert. The secretary responds with a series of prompts. The answers you give in response to these prompts determine what type of analysis FACSDesk will deliver. When the final prompt has been answered, the secretary places a description of the requested analysis onto the white analysis line. This request is for an integration value. Before we explain how to answer the secretary's prompts, let's explore the options FACSDesk offers for analyzing list mode data and discuss some basic data analysis strategies. Several variations of the analysis formats shown in Part 1 are available to you. To display single parameter distributions, you may choose between two different methods of generating histograms. The standard method simply tallies up and plots the number of data points that fall within each brightness range on the x-axis. The density method counts the cells in each brightness range and then uses a density estimation function to plot the curve. Density plots are less sensitive to statistical variation. They tend to reflect fluorescence and scatter distributions more accurately than standard histograms drawn from the same number of cell data points. In addition to selecting the plotting method, you may also select the vertical scale so as to raise or lower the overall height of the curve. 100% is the default scale. Lowering the scale to 50% doubles the size of the peaks. When displaying two-parameter data in the contour map format, there are also several options you may choose from. First, you must select which parameter is to be displayed on each axis. Then you may choose the method of contour placement. Lastly, you may indicate the contour ratio. To explain these options, we'll first review the method in which contour maps are generated. Pairs of measurements from each cell in the data list are plotted onto a two-dimensional grid. This generates a two-parameter histogram in which populations of cells with similar measurements for one or both parameters appear as mountains. To map out these three-dimensional landscapes, FACSDesk draws contour lines which are then projected into a single plane. Two types of contour maps were described in Part 1. Density maps are generated from contour lines drawn in equal intervals from the mountain base to the topmost peak. This places the greatest number of contours around tall peaks, regardless of the number of cells present under the peak. Probability maps are drawn by placing contours so that an equal percentage of cell data points fall between adjacent pairs of contours. These maps offer a view of the data that more accurately reflects the relative number of cells in each region. FACSDesk provides two additional methods for creating contour maps. The logarithmic method places the first contour at a specific height on the mountain range. It then places the remaining contours at logarithmic intervals. For example, a 50% logarithmic contour map would place the first contour at half the height of the mountainscape. The second contour is placed at half the height of the first contour. The next contour is placed at half the height of the second contour, and so on. These maps tend to place many contours around the low lands of a data landscape, so they can be useful for revealing small subpopulations. The area method of drawing contours divides the base of the landscape into concentric regions of equal surface area. In this example, 10% of the area of the map is contained between each pair of contours. These maps tend to heavily emphasize the low lands of a landscape, so that the peaks are effectively lost. The valleys, on the other hand, are clearly defined, so these maps are sometimes useful for establishing cell sorting and integration boundaries. Choosing the appropriate contour map depends on your data and which subpopulations you are interested in revealing. Each type of map has its use. In general, it's a good idea to first call up a probability map so that you can get a clear picture of the overall features in a landscape. Whereas histograms and contour maps graphically display data, integration analyses allow you to present data in tabular form. When requesting an integration, you specify a gate or range of measurements for each parameter. FACSDesk then calculates the percentage of cells that fall within the gating boundaries. Integration analyses also provide mean fluorescence and scatter values for the cells in the gated population. Gating may also be used when generating histograms and contour maps. Tom Kipps used gated contour maps to display the results of his triplet B-cell study. The gating strategy that Tom used is fairly typical. Previous studies in which gated populations were sorted and analyzed by light microscopy showed that lymphocytes tend to scatter less light than other kinds of blood cells. Therefore, as a first step in locating the lymphocyte subpopulations in his samples, Tom examined ungated contour maps displaying forward versus obtuse scatter. He then drew boundaries around the population which he knew to contain lymphocytes, that is, the population which scattered the least amount of light in both directions. Using the scales along the edge of the scatter map, Tom determined the numerical values of the boundaries he had drawn. By entering these limiting scatter gates into FACSDesk, he obtained contour maps showing red versus green staining patterns, mainly for the lymphocytes in his samples. To take data analysis requests, the secretary presents you with a prompt for each FACS sensor in your experiment. To respond to these prompts, you indicate how the data collected from each channel should be handled in the analysis. The secretary uses your responses to appropriately adjust the analysis machine. If you answer the sensor prompts by entering an X-axis for one channel, Y for another, and gates for the remaining channels, the analysis machine will generate a contour map. Entering X for one channel and gates for all others delivers a histogram displaying data from the indicated channel. Lastly, if you enter either an open or limiting gate for each parameter, the analysis machine will calculate the percentage of cells in the gated population. We'll now take a closer look at how to insert integration requests onto an analysis card. To insert requests, the cursor must be at the analysis line level. With the flat commands, position the cursor on a white line below the sample you wish to analyze. Press Insert to begin the analysis prompt sequence. Fluorescein is the first prompt for this experiment. The X which appears in the black box is a default response. To enter an open gate, simply type 0 and press Return. The secretary always assumes the upper boundary of a gate to be 250 unless otherwise indicated. For Texas Red, we enter a lower gating boundary of 110. For obtuse scatter, we enter boundaries of 10 and 100. After answering the prompt for the last sensor, we press Return and the new analysis line appears on the card. On the new line, the gating boundaries for each parameter are listed below the corresponding reagent or sensor heading on the card. The left side of the line is labeled to indicate the type of analysis requested. To review, position the cursor and press Insert. For each sensor prompt, use the main keyboard to enter gating boundaries and then press Return to move on to the next prompt. When you press return after responding to the final sensor prompt, the secretary adds the new analysis request to the card. To request the same analysis to be run on many samples, you can save yourself time and effort by defining a macro command for the analysis. New macros are defined by pressing the Enter key twice in succession and then pressing the key to which the macro will be assigned. In this example, we'll assign a macro to the A key, which will insert a single integration request. To open or begin defining the macro, we press the Enter key twice and then press the letter A. The message Defining Keyboard Macro A appears on the screen, and we begin entering the integration request. We type Insert, then, after entering each set of gates, we press Return. Every keystroke given while defining a macro command will be recorded in the order it is given. When the analysis request is complete, we close the macro by pressing Enter 3 times in a row. This causes the Defining Macro A message to disappear, thus indicating that the macro is complete. If you make an error while defining a macro, pressing the line feed key will cancel the macro definition command, thereby erasing what you have typed so that you may start again. If you wish, you may define recursive macros that call up other macros. Recursive macros often take a very long time to run. Giving the command Control-C will interrupt a macro command before it has completed its task. We can now use macro A to place an integration request for any sample by simply positioning the cursor under each gray line and pressing Enter A. Once you have placed your analysis requests, you can submit the jobs either by checkpointing or by giving the command Enter, Enter, Execute from the analysis line level. When the secretary receives the Enter, Enter, Execute command, it places all jobs requested from that drawer onto the analysis queue. Analyses are normally processed on a round-robin basis in the order that they are received. Unless there is heavy traffic on the system, your results will be returned to your desk quite rapidly. If for some reason you need the results of an analysis immediately, you can submit the job with rush priority status. The priority menu is an optional prompt you may choose from when requesting an analysis. To view optional prompts in the analysis sequence, use the Tab key instead of the Return key to complete your response to each prompt. In the following example, we'll submit an integration analysis with rush priority. We respond to each of the required sensor prompts. Then, after entering a set of gates for the last parameter, we press Tab to progress to the optional priority prompt. We position the cursor next to the rush option, then press Return or Tab to complete the analysis request. When you give a job rush priority, a box flashes around the word queued, indicating that the job is being processed. Only one job at a time may be given rush priority. The results of the normal priority jobs have now been returned to the desk. The number on the right side of each analysis line indicates the percentage of cells in that sample that satisfied the gating conditions of the analysis. The mean fluorescence and scatter values for the cells within the gates can be viewed at the lowest level of the analysis drawer. By positioning the cursor at sample A1 and giving the down-level command, we bring the mean and scale values for the integrated cells into view. By using the flat commands, the cursor may be moved so as to view the statistics on any other sample in the drawer. To request a histogram, you must assign one parameter to be displayed on the x-axis. Here we'll call for a fluorescein histogram. We press Insert, then press Return to accept the X-axis default for fluorescein. To conclude, we enter gates for the remaining channels. To request a Texas Red histogram, we enter gates for fluorescein, press Return to accept the X-axis default for Texas Red, and then enter gates for the remaining sensors. Pressing Return after the final sensor prompt automatically accepts defaults for all optional prompts, thus yielding a 100% density histogram. On a histogram output page, the requested distribution appears in a large box on the lower half of the page. The x-axis is marked with a logarithmic scale, indicating that logarithmic data was collected for Texas Red in this experiment. The upper edge of the box is scaled linearly from 0 to 250. This scale is used for determining gating boundaries. The type of curve is labeled in the upper corner of the chart. The percentage figure indicates the vertical scale used in drawing the curve. In addition to the requested curve, miniature histograms for each of the other parameters appear on the top half of the page. The numbers which appear in the upper corner of a miniature histogram indicate the gating restrictions used on the channel when generating the main curve on the page. If you use Tab instead of Return when answering prompts for a histogram, the secretary will present you with 4 optional menus. You may select between a standard histogram or a density histogram. Density is the default. You may adjust the vertical scale of the histogram. 100% is the default. You may select the graphic medium to be used, and lastly, you may select priority for the job. To request a contour map, you must answer the required sensor prompts by selecting X for one parameter, Y for another, and entering gates for the remaining channels. Pressing Tab to complete each response will advance the cursor to the following optional menus. You can select the contour method, the contour ratio, the graphic medium, and the priority. Pressing Return after answering the final sensor prompt will skip over all optional prompts and yield a 10% probability contour map. The percentage figure is the contour ratio. In a 10% probability map, 10% of the cells in the sample fall between each pair of contours. To see more detail in a probability map, you lower the contour ratio. A 5% probability plot divides the mountainscape into 20 segments of equal cell data volume. The default contour ratio for a logarithmic map is 50%. In this case, raising the contour ratio increases the detail. Watch the screen now as we request a contour map. We enter X for fluorescein, Y for Texas Red, and gates for the scatter channels. Pressing Tab advances to the first optional prompt. Using the word right key, we position the cursor next to the logarithmic option and press Tab again. The secretary prompts us to enter a contour ratio. By pressing Tab again, we accept the 50% default and advance to the graphic medium prompt. We select the plotter and again press Tab, thereby advancing to the priority option menu. When we press Tab to accept the normal priority default, the new analysis line appears on the card. It's important that you familiarize yourself with the format of each type of analysis request. Let's take a look at the variety of analysis requests that have been placed for sample A1A. On the first analysis line is a request for a 100% density Texas Red histogram. The next 4 requests are for different types of red versus green contour maps. The 6th line is an integration analysis that has already been processed. And the last analysis line on the card is for a fluorescence histogram. All of these analyses are gated to exclude cells with high scatter readings. Exiting desk, checkpointing, or pressing Enter, Enter, Execute at the analysis line's level will submit all requested jobs in a drawer for processing. In addition to printing out analyses, FACSDesk also offers several options for printing out the information stored in an analysis drawer. You may request a list of samples in the drawer, a printout of all integrations in the drawer, a list of all curves that have been plotted, or a detailed list of integrations showing mean fluorescence and covariance values. To view these options, position the cursor next to the analysis drawer at desktop level and press Execute. This concludes our introduction to FACSDesk. The information presented in this videotape will get you off to a good start in mastering FACSDesk. As you practice using your desk, you'll find that some commands may only be given at certain levels. If you give a command that cannot be carried out, the terminal will emit a beep. Don't let this alarm you. You cannot damage FACSDesk through the keyboard. The only command to be cautious of is Control-C. If Control-C is given twice in succession, it can interrupt the desk system. If you have any questions while running FACSDesk, use the Enter H command to bring the online manual to the screen.